For decades, the expansion of waistlines in middle age has been dismissed as an inevitable consequence of aging. While lifestyle factors like reduced physical activity and dietary habits have long been blamed, a new layer of understanding has emerged that challenges this conventional wisdom. Recent research has uncovered a biological mechanism that lies at the heart of age-related fat accumulation, particularly around the abdominal region. This discovery not only explains why bellies tend to grow larger with age, but also opens the door to potential therapies aimed at curbing this process and mitigating its harmful health effects. The findings reveal that aging triggers the activation of a previously unknown type of stem cell, which plays a central role in the rapid production of fat cells. These cells, known as committed preadipocytes specific to aging, are highly efficient at generating new fat tissue, particularly in the belly area. The research also identifies a key signaling pathway that drives this process, offering a promising target for future interventions. By delving into the biology of these cells and their role in metabolic health, scientists have taken a significant step toward addressing one of the most pervasive health challenges of modern life. At the core of this groundbreaking discovery is the identification of a novel type of stem cell that emerges during middle age. These cells, referred to as committed preadipocytes specific to aging, represent a subset of adipocyte progenitor cells, APCs, which are responsible for creating new fat cells. Unlike their counterparts in younger individuals, these age-specific cells exhibit an unprecedented level of activity, rapidly multiplying and evolving into fat cells. This heightened capacity to produce fat contributes significantly to the expansion of white adipose tissue, the type of fat associated with weight gain and metabolic disorders. What makes these cells particularly intriguing is their unique behavior compared to other adult stem cells. While most stem cells lose their regenerative potential as the body ages, committed preadipocytes defy this trend. Instead, they become more active with time, transforming into prolific fat-producing factories. This phenomenon helps explain why older individuals often experience an increase in body fat even when their weight remains stable or their muscle mass declines. The emergence of these cells marks a pivotal shift in how the body handles fat storage, turning what was once a controlled process into a runaway mechanism of fat accumulation. A critical aspect of this discovery is the role played by a specific signaling pathway, known as leukemia inhibitory factor receptor, LFR. This pathway acts as a catalyst, driving the transformation of committed preadipocytes into fully formed fat cells. In younger organisms, the LFR pathway appears to be less essential for fat production, suggesting that its activation is closely tied to the aging process. As individuals grow older, the LFR pathway becomes increasingly important for the proliferation and differentiation of these fat-producing cells. This insight provides a clear target for potential therapeutic interventions aimed at disrupting the excessive fat production seen in middle age. To validate their findings, researchers conducted a series of experiments using both animal models and human tissue samples. Initial studies focused on mice, where adipocyte progenitor cells were isolated from both young and older animals. When transplanted into younger hosts, cells from older mice demonstrated a remarkable ability to generate vast amounts of fat tissue. In contrast, cells taken from younger mice exhibited minimal fat-producing activity, regardless of the age of the host. These results underscored the intrinsic changes that occur within adipocyte progenitor cells as they age, highlighting their newfound capacity to independently drive fat accumulation. Further investigation involved the use of advanced techniques such as single-cell RNA sequencing, which allowed researchers to examine gene activity at an unprecedented level of detail. Comparing adipocyte progenitor cells from young and older mice revealed striking differences in their behavior. In younger animals, these cells remained largely dormant, with minimal activity related to fat production. However, in middle-aged mice, the same cells became highly active, churning out new fat cells at an accelerated rate. This dramatic shift provided further evidence of the biological changes that underpin age-related fat gain. To confirm the relevance of these findings to humans, researchers turned their attention to adipose tissue samples obtained from individuals of varying ages. Using the same analytical methods, they identified similar patterns of activity in human adipocyte progenitor cells. Just as in mice, these cells showed little activity in younger individuals but became highly engaged in middle age, producing large numbers of fat cells. This parallel between species strengthens the validity of the findings and underscores their potential implications for human health. While the cosmetic impact of belly fat is often the first thing people notice, its effects on health are far more consequential. Excess abdominal fat is not just a superficial concern, it plays a central role in accelerating aging and disrupting metabolic processes. Visceral fat, the type that accumulates deep within the abdomen, is particularly harmful because it surrounds vital organs and releases inflammatory substances that contribute to chronic disease. This type of fat is strongly associated with an increased risk of conditions such as type 2 diabetes, cardiovascular disease, and certain cancers. One of the key ways belly fat exerts its detrimental effects is by slowing down metabolism. As fat cells multiply and expand, they interfere with the body's ability to regulate blood sugar levels and burn energy efficiently. This metabolic dysfunction creates a vicious cycle, making it harder to lose weight and increasing the likelihood of developing insulin resistance, a precursor to diabetes. Furthermore, the inflammatory compounds released by visceral fat can damage blood vessels, raising the risk of heart attacks and strokes. The connection between belly fat and aging is particularly concerning. Fat accumulation in the abdominal region is not only a marker of aging but also a contributor to it. The presence of excess fat accelerates cellular aging and promotes oxidative stress, further exacerbating the decline in metabolic health. By identifying the mechanisms behind this process, researchers have taken a crucial step toward breaking this cycle and improving health outcomes for aging populations. The identification of committed preadipocytes and the LFR signaling pathway as key players in age-related fat accumulation offers exciting possibilities for future therapies. One potential approach involves targeting these cells directly to reduce their activity or eliminate them altogether. By blocking the LFR pathway, it may be possible to prevent the transformation of committed preadipocytes into fat cells, effectively halting the excessive production of belly fat. This strategy could provide a novel way to address obesity and its associated health risks without relying solely on lifestyle modifications. Another avenue of exploration involves developing drugs or treatments that specifically inhibit the proliferation of committed preadipocytes. Such therapies could be tailored to individuals at risk of age-related fat gain, offering a personalized approach to managing metabolic health. Additionally, researchers are investigating whether interventions that target these cells could extend health span, the period of life spent free from chronic illness, by reducing the burden of conditions linked to excess fat. Future studies will focus on tracking the behavior of committed preadipocytes in living organisms and observing how they respond to various interventions. Longitudinal research involving human participants will be essential to determine the effectiveness of these strategies and their broader applicability. By combining insights from animal models with data from human studies, scientists hope to refine their understanding of these cells and develop therapies that can make a meaningful difference in the lives of millions. This research represents a significant advancement in the field of longevity science, which seeks to understand and counteract the effects of aging on the body. By uncovering the biological mechanisms behind age-related fat accumulation, scientists have identified a critical piece of the puzzle that determines how we age. The discovery of committed preadipocytes and their role in fat production highlights the complexity of aging and underscores the importance of targeting specific cellular processes to improve health outcomes. As the global population continues to age, finding ways to mitigate the effects of aging on metabolic health will become increasingly urgent. The insights gained from this study offer hope for addressing one of the most pervasive health challenges of our time. By targeting the root causes of age-related fat gain, we may be able to not only extend lifespan but also enhance quality of life, allowing individuals to enjoy more years free from the burdens of chronic disease. In conclusion, the identification of committed preadipocytes and the LFR signaling pathway marks a turning point in our understanding of age-related fat accumulation. This breakthrough paves the way for innovative therapies that could transform how we approach obesity, metabolic health, and aging itself. With continued research and development, the dream of a healthier, longer life may soon become a reality for many. And there you have it, science just uncovered one of the biggest mysteries behind why our waistlines expand as we age. This isn't just about vanity, it's about understanding the biology of aging and how our bodies change in ways we never expected. The discovery of these fat-producing cells and the signaling pathway driving them gives us hope for new solutions to combat belly fat, improve metabolic health, and maybe even extend our healthy years. If you found this video insightful, don't forget to give it a thumbs up and subscribe for more deep dives into the latest science and health discoveries. Thanks for watching.